Hello everyone, Mr. Linder here. Let's talk about cellular mechanisms of action for water-soluble hormones. So in this video, I want to talk about how water-soluble hormones can interact with receptors in the plasma membrane of cells and go on to exert cellular responses. So most of your water-soluble hormones are going to be peptide hormones. Now, there are some examples when you get into thyroid hormone mechanism where you have an amine-derived hormone that can work through a different mechanism. But most of the time when you're talking about peptide hormones, you're talking about hormones that are lipophobic. And so they don't want to pass through the plasma membrane or can't pass through the plasma membrane. And so what these hormones do is they interact with receptors that are found within the plasma membrane. And so in these generalized examples, you have hormones interacting with a receptor. And in this case, it's going to activate something called a G protein. And so we'll talk about that mechanism. And then that G protein can go on to activate an enzyme. And then that enzyme can do work within the cell through things that are called second messengers. In this generalized example, the hormone's binding to a receptor and it's activating an enzyme known as a tyrosine kinase. And again, we can go on to activate proteins within the cell, and we can eventually have what we refer to as the cellular mechanism of action. The cell can respond and actually have an opportunity to do physiological work. So the examples I want to go through when we look at predominantly peptides, and we're again talking about water-soluble hormones, so things like insulin or follicle-stimulating hormone, luteinizing hormone, growth hormone, etc., is we want to look at mechanisms called the cyclic AMP second messenger system, the tyrosine kinase second or messenger system, and the phospholipase C messenger system. So I want to look at these three mechanisms to give us a cellular mechanism of action. What's interesting about all of these mechanisms is that they can give you quite a rapid response as it relates to hormones. Hormones are a slower process in general, but because the machinery is already built into the cell, we can get a faster response because of the existing receptors, the existing G proteins and enzymes within the membrane, and of course the existing proteins within the cell that can go on to do work. So really we're looking at cellular work being done in the cellular response to the hormone. So let's look at the cyclic AMP second messenger system. So in the cyclic AMP second messenger system, you're going to utilize what's known as a G protein. And the G protein actually consists of subunits. So when you look at a G protein, you're going to find that there is an alpha subunit, and then there's a beta and gamma subunit. So the G protein is attached to the receptor. So GPCR stands for G protein coupled receptor. So what we have here is a receptor, the receptor, which is interacting with a hormone. So a hormone's going to bind to the receptor, and it's going to go through an activation of a G protein. And the G protein, again, is made up of subunits, alpha, beta, and gamma. When you activate the G protein, you can go on to activate an enzyme that's found within the plasma membrane, adenylyl cyclase or adenylate cyclase, often just abbreviated as AC. And that adenylate cyclase is important because it allows us to produce cyclic AMP. And so cyclic AMP becomes the second messenger within the cell. The first messenger being the hormone that's binding to the receptor, which is outside of the cell. It's not able to pass through the plasma membrane. So here we have the plasma membrane. The hormone's not coming through the plasma membrane. Instead, it's binding to a receptor in the membrane. And we're generating a second messenger that's found within the cell that then can go on to do work. So what does the mechanism look like? What are the steps if we were going to walk through the process? So number one, the first thing that you want to consider is that the hormone is binding to a receptor. So we have an interaction of this water-soluble hormone with a receptor in the plasma membrane. The second thing that we see happening is the G protein is going to split into two parts. We're going to get an alpha subunit and we're going to get a beta gamma subunit. So if we were to draw an alternate picture here, so we have our plasma membrane and we have our receptor and we have our hormone that has attached to the receptor, and then we have our G protein. So we've got the alpha and then we've got the beta gamma. I'm going to draw it like this so that you can see the two parts here. When the receptor activates the G protein, what's happening is these subunits are separating. So if you can imagine in the membrane, you'll have an alpha and then you'll have a beta gamma subunit. So they've actually split apart from each other. And when they do that, you actually drop off energy in the form of GDP. So GDP is a lower energy form than GTP. So we lose GDP in the process of splitting that G protein. Depending upon the tissue type, so depending upon where you're located in the body, sometimes the alpha will continue on through the plasma membrane, or sometimes it's the beta gamma that will move through the plasma membrane. So you might have the alpha continue on through the plasma membrane, or in a different tissue, you might have the beta gamma continue on through the tissue. It doesn't really matter when you're first learning the mechanisms. You just want to understand that one of the subunits is going to pick up energy and it's going to move through the plasma membrane. So let's say, for example, that the alpha subunit is going to pick up energy and it's going to move through the membrane. So let's go back to our original diagram here. Let's say that the alpha subunit is going to move through the membrane, and that's the one that's going to move on and activate the enzyme. That subunit will attach to itself GTP. So the alpha subunit will pick up GTP. That's a higher form of energy. And that alpha subunit will leave behind the beta gamma, and it'll move through the plasma membrane and attach to an enzyme called adenylate cyclase, or also known as adenylyl cyclase. So this is the enzyme embedded in the plasma membrane that's going to interact with the active subunit. So in this example, we're saying that the alpha picks up GTP and it's moving through the plasma membrane and it's binding to the enzyme. This enzyme then becomes active when the active subunit is attached to it. So the alpha with the GTP comes and binds to the enzyme. And what that does is it turns on the enzyme and allows it to do its job. The job of the adenylate cyclase is to take ATP and convert it into cyclic AMP. So we are producing cyclic AMP from ATP. Once you have cyclic AMP, that becomes the second messenger. So cyclic AMP is now this second messenger inside of the cell. And it can then go on to perform other tasks within the cytoplasm. And one of the tasks that cyclic AMP has is to go and turn on an enzyme known as protein kinase. So cyclic AMP is going to come along and it's going to activate. So it's showing you three examples here. Cyclic AMP coming along and it's activating protein kinase. Kinases are in the family of enzymes that phosphorylate. Okay. So if you were going to go back and sort of number this, number 1 was hormone binds receptor. Number 2 is the G protein dissociates. Number 3, the active portion goes through the membrane. Number 4, we're going to activate adenylate cyclase. And now we're on to this idea of number 5. The cyclic AMPs are interacting with the protein kinase. And protein kinases, all kinases are phosphorylators. Okay, so they phosphorylate. What does that mean? It means they add phosphates, and we understand that phosphates are really energy. So really what a protein kinase is doing, whether it's protein kinase A or whether it's protein kinase C, whichever version it is, it's adding energy so that other proteins within the cell can go on to do work. So the protein kinase is going to add energy. Okay, so I can write right here, it's adding energy. Okay, it's phosphorylating all these other proteins. Okay, so see all these other proteins within the cell. Okay, these protein kinases are going along and they're adding energy to these proteins so that those proteins within the cell can elicit the cellular response. So these proteins now that have phosphates attached to them, they've been energized, can go on and produce the cellular response. And what you should notice through this mechanism is that we are amplifying the response. We started with one hormone, right? So we start with one hormone binding to a receptor, and we produce lots of cyclic AMP, and so we activate lots of protein kinases, and then we go on to phosphorylate and activate lots of proteins within the cell. And so you can see the cellular response becomes bigger than just the one hormone at the beginning. We get this amplification process from that single hormone. This is why hormones can be at small concentrations. You can have nanomolar concentrations of a hormone, but you can get a larger cellular response because of this amplification that takes place within the cytoplasm. And so once these proteins are phosphorylated, then the cell can change the permeability of ion channels. It can modify the activity of enzymes within the cell for metabolism. It can change the transport proteins within the plasma membrane, and so forth and so forth. So depending upon the hormone and depending upon what cells in which particular tissue within the body, you can get differing types of cellular responses. So then you can go back and you can talk about what are the effects of follicle stimulating hormone that might use this mechanism, or what are the effects of a growth hormone that might use this mechanism, and so forth and so forth and so forth. So this is an example of a mechanism that can be utilized by different types of hormones to give you a cellular mechanism of action. To turn this off, you would break off a phosphate from the G protein subunit. So we're going back to the alpha subunit with its GTP. You can actually break off a phosphate and return this alpha into the GDP form. Again, imagine this would be within the plasma membrane, not down here in the cytoplasm. So this is taking place in the plasma membrane. And then this subunit can move back through the membrane, combine back with the beta gamma, and reattach to the receptor, and then this process could go again. So if we were to draw this, it might look something like this, where the alpha subunit with its GTP is attached to the adenylate cyclase. And so what we're doing is we're turning this off by breaking off a phosphate. So the alpha now has GDP attached to it, and it's moving its way back through the plasma membrane. It's going to link up with the beta gamma subunit. So they're going to combine back together. So now we have alpha, beta, gamma as the G protein. It has that lower energy GDP attached to it. And then it's going to go back and reattach with the original receptor. And then you could start this process all over again. A hormone can come along and bind with that receptor, activate the G protein, and start the process going back through the membrane again. So this is the cyclic AMP second messenger system. And it turns out there's another second messenger system which is exactly like this one. It just uses a different type of enzyme in the membrane. It's referred to as GC, or guanylate cyclase, and it produces cyclic GMP. But the mechanism would be the same. So whether you're making cyclic AMP or cyclic GMP, it just depends on which enzyme you're using in the membrane, but the mechanism itself is still the same. What's also interesting is there are clinical applications to this as well. Before we take a look at that, I want to show you just a more complex diagram. This can be overwhelming at times, but what this diagram is showing you is that you have plasma membrane with lots of different types of receptors and lots of different chemicals that can interact. So whether it's calcium affecting receptors or channels, whether it's hormones binding to receptors that are attached to G proteins. And so here's the alpha subunit, and then here's the adenylate cyclase in the membrane. You have lots of different mechanisms that are taking place within a cell all at the same time. I like this one though because you can see the adenylate cyclase, and then if we go here, you can see the production of cyclic AMP, and then you can also see that cyclic AMP is going to go on and affect protein kinases. So we can follow this down here. So here we have protein kinases within the cell, and then those can go on to affect lots of different proteins within the cell, and you can get lots of varying types of responses. If you're going to turn this system off, you need an enzyme that can degrade cyclic AMP. So this enzyme right here, PDE stands for phosphodiesterase. And phosphodiesterase is used to break down, it's the enzyme that breaks down cyclic AMP. And so if you can break down cyclic AMP, you can shut off the second messenger within the cell. So that's a really important enzyme because it's going to turn off cyclic AMP. So just like you have degradation of hormone on the outside of the cell, you need degradation of the second messenger within the cell in order to shut the system down. But clinically, we can modify what the phosphodiesterase is doing, and we can change the activity of a particular cell for a clinical benefit. So for example, there are drugs that are phosphodiesterase inhibitors. So there are PDEIs, the I representing inhibition. So there are phosphodiesterase inhibitors that can allow for levels of cyclic AMP to stay elevated within a cell and give us a clinical physiological response that we're going for. So for example, a drug like theophylline. Theophylline is a phosphodiesterase inhibitor, and what it does is it allows for an increased cyclic AMP levels within bronchial smooth muscle, and theophylline can go on to bronchodilate. And so you might see this used in patients that have asthma, where you need to bronchodilate, you need to relax the smooth muscle, and then allow for bronchodilation to aid in breathing. We also see this with caffeine. Caffeine is actually a phosphodiesterase inhibitor in certain areas of the body. Caffeine in a pinch can actually be used to treat somebody with an asthma attack. It's not as good as something like a theophylline. But it is a minor stimulant, and it can be used to allow for bronchodilation as well. We also know caffeine has effects in the central nervous system as well. It actually changes the levels of dopamine available, which is why it has that stimulatory effect, and also why we have caffeine addictions as well, because it's tied to dopamine release. Another example of a phosphodiesterase inhibitor is sildenafil. You might know this as Viagra. And so it too can affect the levels. I'm going to jump ahead here. It can affect the levels of cyclic AMP. We can elevate cyclic AMP levels through phosphodiesterase inhibition. We can see that relaxing musculature in the erectile tissue, and that can allow for the treatment of infertility issues, so erectile dysfunction. But all of these are similar because they are inhibitors to phosphodiesterase activity. And so they increase those cyclic AMP levels to give us the response we're looking for. And so oftentimes it's relaxation of smooth muscle, or it could be somewhere else as sort of treating inflammatory things, whatever. It could be a lot of different examples, but I wanted to show you these smooth muscle examples. If we go back, though, I wanted to show you this example. Adrenaline, noradrenaline, what we know as epinephrine, or norepinephrine. These actually work through various receptor pathways, alpha-1 receptors, alpha-2 receptors, beta-1, beta-2, all kinds of different varieties of receptors for epinephrine and norepinephrine. Notice that they work through multiple pathways, phospholipase C, but these are the adenylate cyclase pathways. And so you can see that using drugs can interact with various types of mechanisms, and we can control, again, here's smooth muscle relaxation, but it also may be tied to muscle contraction in the heart or some metabolic event. And so we can manipulate what the cellular physiology that's taking place based on the cellular mechanism of action of a particular hormone, a particular chemical. And in the nervous system, you know that epinephrine and norepinephrine can act as neurotransmitters, but they also act as hormones as well. Another cellular mechanism of action is the phospholipase C mechanism. So PLC for short. And the phospholipase C mechanism is also a GPCR. It's also a G protein coupled receptor mechanism. In the phospholipase C pathway, the big difference here is that you're activating a different enzyme. So you're activating the enzyme phospholipase C, and you're also going to see changes in calcium levels. So we're actually going to be using calcium as one of the messengers inside of the cell. So what would this particular water-soluble hormone mechanism look like? So it's just a different example of a cellular mechanism of action for a water-soluble hormone. And so something different than the adenylate cyclase pathway. This is the phospholipase C pathway. In this particular pathway, it still starts out the same. Number one, hormone would have to bind to a receptor. So hormone's going to interact with a receptor. And then, of course, number two, it's going to activate a G protein. So if we were going to say the second step here is activation of a G protein. And again, the G protein is made of three subunits, alpha, beta, and gamma. So everything is still the same. And of course, you have that GDP attached to it that's going to fall off once it gets activated. Step number 3, the active subunit is going to move through the membrane. So let's just say, for example, in this one, it's the beta gamma that becomes activated. And so when it's active, it'll have GTP attached to it. So that active subunit is going to move through the membrane. Hormone binds receptor, activates G protein, active subunit. So number 3, active subunit moves through the plasma membrane, and it's going to interact with phospholipase C. That is the enzyme that's in the plasma membrane. If you get lost, there's a key on this particular diagram, and so here's, because of all these acronyms, here's PLC and phospholipase C. So all these acronyms have names for them. So what does the phospholipase C do? Well, this particular enzyme is going to take a phospholipid. So our cell membranes are made of phospholipids. It's going to take a particular phospholipid. It's called PIP2, PIP2. It's going to take PIP2 and it's going to convert it into 2 molecules. So the PIP2 is going to be converted into IP3 and DAG. So these 2 molecules, IP3 is inositol triphosphate and DAG is diacylglycerol. So we're making 2 molecules from a phospholipid that's inside of the plasma membrane. And it's all because we've activated the phospholipase C enzyme. What does diacylglycerol do? So this would be step number 4, making these 2 molecules. Step number 5 would be diacylglycerol is going to go on and it's going to activate protein kinase. So here we see a different kind of protein kinase. This is protein kinase C, but all kinases are doing the same thing. So they are phosphorylating. Kinases add energy. So we see the protein kinase adding energy to proteins within the cell so that they can go on to perform their cellular response. What about IP3? So let's call this number 6. Okay? IP3 is going to go on and it's going to interact with the endoplasmic reticulum. So IP3 is going to come along and it's going to bind to receptors that are in the membrane of the endoplasmic reticulum. And when IP3 interacts with its receptor, it's going to cause the release of calcium. So what we see happening is calcium gets released as a messenger, right? So it's acting as another messenger inside of the cytoplasm, inside of the cell. So we have DAG acting as a messenger, we have IP3, inositol triphosphate, acting as a messenger, and now we have even calcium acting as a messenger inside of the cell. That calcium is actually going to go on to interact with a protein called calmodulin. So calcium interacts with calmodulin, and that calcium calmodulin is able to actually turn on protein kinase as well. So this diagram doesn't show that, but we're going to go and add this in here. So calcium interacts with calmodulin, it interacts with protein kinases, and we know protein kinases are, again, phosphorylators. So what the protein kinase is doing is it's adding energy, it's phosphorylating, so we get phosphorylation, so that you can have ultimately the cellular response from the initial hormone. So whatever hormone is utilizing this pathway, this cellular mechanism of action, whatever hormone is binding to the receptor, it's going to go through this series of activations so that you can get ultimately the cellular response. And so this is known as the phospholipase C pathway. The last main pathway for water-soluble hormones is the tyrosine kinase pathway. The tyrosine kinase pathway is a little bit different. In this mechanism, what you're actually going to see is an activation of the receptor, and then the receptor is going to go on to phosphorylate proteins within the cell. So from a basic standpoint, what you're seeing here is a hormone So here it's listed as ligand, okay? But hormones are ligands, okay? So the hormone is binding to the receptor, and then you're getting activation of the enzyme. So the enzyme that we're talking about is tyrosine kinase. So tyrosine kinase is the enzyme. And because it's a kinase, we've learned that kinases add energy. So they're able to phosphorylate. They're able to add energy to proteins so that these proteins can go on to do work within the cell, and so we get a cellular response. So through this tyrosine kinase mechanism, what we're trying to accomplish is the same thing. We're trying to get a cellular response from a hormone that binds to a receptor that's located within the plasma membrane. So we're still talking about water-soluble hormones. The best example of this is insulin. Insulin is a great example of a hormone that utilizes the tyrosine kinase mechanism, and other insulin-like growth factors will utilize this particular mechanism. So let's look at it a little bit closer. So what's really happening in this particular mechanism? So what we see actually in the plasma membrane is the receptor starts out as two separate pieces in the plasma membrane. So this is actually one half of the receptor. This is half of the receptor here, and this is the other half over here of the receptor. So insulin, as an example, the hormone, comes along, and it's going to cause what we refer to as dimerization of the receptor. So the tyrosine kinase receptor exists as two separate parts. So when the insulin comes along, it's going to bring those two halves together. And so what we can see is when those two halves come together, we get what's known as dimerization. So now we have the full receptor. Now that they are attached to each other, this whole structure, okay, dimerization, this whole structure is the receptor. But the receptor can only come together if the hormone is present. So we have the hormone interacting on the outside of the cell. And then we have the internal portion of the receptor, which is going to be the enzyme part. So the outside portion is actually interacting with the hormone, and the internal part is actually going to be the enzyme. The next thing that we see happening is what we call autophosphorylation. What happens is the enzyme portion of the receptor is going to take phosphates. So here's a phosphate here and a phosphate here. It's going to be taking phosphates from pre-existing energy within the cell, ATP. And so we're going to create ADP, and we're going to take off a phosphate, and we're going to attach it to the enzyme portion here. So you have actually phosphorylation of the tyrosine kinase enzyme. And what that does is that makes the enzyme active. So the tyrosine kinase is now active within the cell when it becomes autophosphorylated. So it adds phosphate to itself. Once the enzyme becomes active, then it can go on to do the function of a kinase. And we know that kinases are going to be adding phosphates. So here we have adding a phosphate to other proteins within the cell. So we're going to add phosphates to other proteins, and we know that those proteins are going to go on to do work, right? They're going to go on to do the cellular response. And so in the case of insulin, that could be to insert glut transporters into the plasma membrane. And so we have the cellular response that takes place based on the hormone that attached to the receptor. So tyrosine kinases can stimulate glycogen production, they can stimulate fat production, protein synthesis, they can insert glut carriers into the plasma membrane, all the different types of actions that you might associate with a hormone like insulin. So insulin has lots of different effects, and so its cellular mechanism of action through the tyrosine kinase pathway allows us to see those responses. So what would this look like from just a basic schematic? You have the insulin hormone activating the receptor. So here we see the two halves of the receptor coming together. We see the activation, the phosphorylation of signaling molecules that are going to communicate with vesicles in the cell so that they can move those vesicles out to the plasma membrane, and we can insert glut transporters so that glucose can diffuse into the target cell, because that is one of the actions of insulin, to communicate with cells and essentially tell them to take glucose from the bloodstream to the inside of the cell. Now, of course, this is more complicated than that. So if we look at a more complicated version of this, we can still see the same kinds of things. So here's insulin. This is the hormone at the top up here. This is the receptor in purple. And we can see the phosphates, so the autophosphorylation that took place. And then we can see then the activation of other proteins. So here we have a protein that becomes activated, and it's going to go and affect the vesicles that have the glut transporters. And so we can see those transporters being inserted into the plasma membrane. But there are lots of other pathways that can be affected. So depending upon what proteins get phosphorylated, you can see that there can be other responses that take place. So for example, the pathway might be to activate the enzyme glycogen synthase. Glycogen synthase is what allows us to produce glycogen from excess glucose. So when you have excess glucose entering into a cell, we're going to activate glycogen synthase so we can produce glycogen within the cell and we can store that excess glucose. But we also have pathways that allow us to produce fats as well. So if you take in excess sugars into cells, you know that you can also produce fats as well. Cells can perform lots of different physiological mechanisms based on the receptor that's been activated, and then the different proteins that are activated and the different pathways that become active within the cell. And so insulin can do lots of different things through a tyrosine kinase pathway. So what we just went over are the three main water-soluble pathways. When hormones cannot enter into a cell, they want to bind to, or they need to interact with receptors that then allow you to activate other proteins within the cell, and you can go on to do cellular work. So we looked at cyclic AMP, phospholipase C, and the tyrosine kinase mechanisms. I hope that helps. Take care.